Bismillahirrahmanirrahim, and a very good morning. Hope you all will be fine and in best of your health. I hope you all are availing this remote learning session. Today we will start chapter 3, that is enzyme. Dear students, we are skipping the chapter 2, that is biological molecule, because it is complicated one and inshallah we will do it later. Before going on the topic, first you should know about what are the objectives of our today's lecture. First, we will discuss the definition of enzyme. Then we will move on properties of enzyme. Then the structure of enzyme. Then we will discuss the mechanism of enzyme action, model of enzyme action, and energy activation. So let's start our lecture. As all of you know that number of chemical reactions are taking place in our body, and some of all these chemical reactions occurring in the body is called metabolism. When the word metabolism is used, it is referred as catabolism and anabolism. Now what is catabolism? Catabolism is the breaking down of compound with release of energy. Here you can see that large molecules are converted into small molecules with release of energy. And when we talk about anabolism, what is anabolism? Anabolism is the building of compounds by using energy. In this process, the small molecules are combined to form the large molecules with using energy. So there are chains of reactions going in the cells called metabolic pathways. These reactions are carried out very quickly so that the product can be utilized in various life activities. So now the question is that how these reactions are carried out? Who are helping them to occur so quickly? So the answer is enzymes. Basically enzymes help them. Now we will discuss the definition of enzyme. Enzymes are thermolabile, biological catalyst, protein in nature, and they speed up biochemical reaction without being consumed. Now let's discuss the different terminologies used in this definition. The first one is thermolabile. This is thermolabile. Now what do you mean by thermolabile? Basically the thermolabile means that these enzymes are sensitive to heat. Or we can say that these enzymes can lose their characteristics property on heating. The next point is, or terminology used in this definition is biological catalyst. As all of you know that what are catalyst? Catalyst is a substance that increases the rate of chemical reaction without itself undergoing any permanent chemical change. And enzymes are called biological catalyst because all reactions in living things depends on enzyme. So the next is about the chemical nature of enzyme. Chemically they are protein in nature. And the next thing is that they speed up the biochemical reaction without being consumed. Their function is that they speed up the chemical reaction and they are not utilized in that reaction. In this figure you can see that here is the enzyme. This is called as the active site of enzyme, and here the substrate, or you can say that reactant comes and combine with this enzyme. Now we will discuss the properties of enzymes. Enzymes are organic catalyst. As all of you know that what are organic? Those molecules which are containing hydrogen, carbon as major element. And some of enzymes act as inorganic catalyst. Enzyme increase the speed of chemical reaction by 10 to the power 6 to 10 to the power 14 times faster than the rate of uncatalyzed reaction. Here in this image you can see that what are enzymes made up of. Basically they are protein in nature, then organic catalyst. Now what is organic? Contains carbon and hydrogen, and water catalyst that affect the rate of the chemical reaction. Now the next property is enzymes are highly sensitive to pH and temperature. How they are sensitive to pH and temperature? They are sensitive to pH and temperature because it can affect the interaction that holds the part of the protein chain in place, or they may lose their shape. So the next point is enzymes are either highly specific or less specific that can catalyze the related reactions. Now what do you mean by enzymes are highly specific? In some reactions they are highly specific. For example, in sucrase, the enzyme sucrase can only digest sucrose. It is its specificity. So the next is slightly less specific. Means वो सिर्फ एक काम नहीं कर रहा, सिर्फ एक चीज़ के लिए specific नहीं है. Their example is carbonic anhydrase can add oxygen to hemoglobin and can control the formation of carbonic acid and bicarbonates in blood. Next point is enzyme work in vivo, means when enzymes are working in living cell. Here in this image you can see that if enzymes are working in plants, in animals, or in human being, they are said to be that they are working in vivo, within the cell, and as well as in vitro, means in laboratory, in industry. Here you can see in the image lipase and proteases in laundry detergent, or you can say that making cheese is the example of in vitro. Now the next property is some enzymes require cofactor for proper activity. Now the question is, what are cofactors? Later on we will discuss it briefly, what are cofactors. Next is enzymes are required in very small quantity for the reaction. Enzymes lower the need of activation energy. Now the question is, what is activation energy? If we simply say that activation is basically speed bump, you can say that it is the minimum energy which the reacting species must have in order to start a specific reaction. Or we can say that this energy is needed to break down the chemical bonds, allow the reaction to occur when an enzyme binds to the substrate molecule to react to form products. Enzyme only speed up a reaction and do not affect the equilibrium of reaction. Means it only gives speed up the reaction, it only starts the reaction and do not affect the equilibrium of reaction. Now what is equilibrium? Equilibrium of reaction means when reactant and product are present in concentration which have no further tendency to change with time. In this image you can say that you can see that there are different properties of enzyme. Now the structure of enzyme. Enzymes are globular protein, or they can also say as spheroprotein. Means they are spherical in shape and usually water soluble. Enzymes are made up of one or more polypeptide. Polypeptide chain are polymers of amino acid joined together with peptide bond. Ribozyme, these are the special type of enzymes. Ribozymes are the enzyme which consist of RNA and are found in ribosomes, work together to produce protein. Now ribozyme is a RNA enzyme that catalyzes a chemical reaction. This ribozyme is also called as catalytic RNA. They are found in ribosome. Basically, it is found in ribosome where they join amino acid together to form protein chain. So here is the structure. Now we will talk about the shape. The molecular weight is ranging from about 10,000 to over million. Such enzymes have tertiary and quaternary structure. Now what is tertiary and quaternary structure? Basically these are the structures of protein. Here I have mentioned that these are structural conformation in protein, means how they are forming, how the proteins are formed. Inshallah, in chapter 2 biological molecules there is a detailed topic in which we will discuss about the shape of protein. There are 4 types: primary, secondary, tertiary and quaternary. In enzymes there are tertiary structure and quaternary structure. You can say that here are polypeptide chain and here are assembled subunits. Now the structure, that is active site. Now what is active site? Active site, the catalytic activity is restricted to active site. The question is that what is active site? Basically this active site is a region of an enzyme where substrate molecules bind and undergoes a chemical reaction. It is 3-dimensional and charge bearing cavity. The reactant is called as substrate. Substrate molecule is attached to an active site by non-covalent interaction. In this image, you can see that there is an enzyme and this portion is basically active site. When a substrate or any reactant combine and join here at this portion at active site, it will form a substrate enzyme complex. Now the active site consists of 3 to 12 amino acids which may be scattered in polypeptides but are bring together in particular fashion due to secondary and tertiary folding of protein molecules. They are present in a specific pattern due to the presence of these protein structures. Which one? That is secondary and the tertiary folding of protein molecule. Now what are the examples? The active site for aldolase. Aldolase is an enzyme and the example of their active site is glycine, histidine, alanine, amino acid. So here is the enzyme and this 3-dimensional structure, it is basically active site. Now this substrate or the reactant comes and combine with enzyme at this position. When this substrate combine with enzymes, it will become enzyme substrate complex. Now something more about active site. What are active site? Active site consists of basically 2 functional regions. The one is binding site and the other one is catalytic site. The active site consists of amino acid residues that form temporary bonds with substrate, which is known as binding site. Binding site basically is that portion where the bond is temporary bond with substrate, and residues that catalyze a reaction of the substrate is catalytic site. Shape of active site is designed according to substrate. हमारे पास जैसे substrate होगा वैसे ही हमारे पास क्या होगी, वो active site होगी. Active site के जो two functional group हैं like binding site and catalytic site, उसको इस image से हम लोग थोड़ा सा समझ लेते हैं. Now this is enzyme and here is active site, and we are saying that active site is divided into two functional region. The one is binding site. Now what is the function of binding site? Binding site holds the substrate in place, and the second one is catalytic site where the reaction occurs. Now the next is active site. Here is a flow sheet diagram. Here is active site. Active site can be further divided into two functional region. The one is binding site and the other one is catalytic site. Now what is the function of binding site? It chooses the substrate and binds it to active site. The next is catalytic site. It performs the catalytic action of enzyme. Now we will move further, and here is the next terminology, that is cofactor. Now what are cofactor? Cofactor is basically non-protein chemical compound, or it may be metallic ion, that is required for the enzyme's activity as a catalyst, a substance that increase the rate of chemical reaction. Here इस catalyst को यहाँ पे explain किया गया है basically. The cofactor can be considered as the helper molecule that assist in biochemical transformation. Now these cofactors are basically non-protein chemical compound. Here in this figure you can see that here is the enzyme, okay, and here the cofactor will attach. In the next slide it will be more clear. On the basis of cofactor, there are two types of enzyme. The one is apoenzyme and the other one is holoenzyme. Now what is the difference between these two enzymes? First we will discuss apoenzyme. What is apoenzyme? An inactive enzyme without the cofactor is called an apoenzyme. Enzyme which is inactive, जो कि अभी काम नहीं कर रहा, जो कि अभी कोई function perform नहीं कर रहा, without the cofactor, and there is no cofactor attached with that enzymes. Such enzyme is known as apoenzyme. Here is the example. Now the next is holoenzyme. An enzyme which requires a cofactor become active only if the cofactor is combined with it. Such an active enzyme is called holoenzyme. Holoenzyme is opposite of apoenzyme. Now in this figure you can see that this is enzyme and it is inactive protein, means it is inactive enzyme. What is the reason that it is inactive? Because there is no cofactor. Okay, but as soon as there is cofactor, it comes and attached at this site, then it will become active protein. Without the cofactor attached, the protein is not active. But as soon as the cofactor binds, a cofactor binding activates the protein. जैसे ही यहाँ पे आके cofactor bind करेगा, at that time protein will become active. Now there is an example of apoenzyme and holoenzyme. Now the pepsin is an example of such enzyme. It is created by gastric gland from stomach wall in an inactive state, and that inactive state is pepsinogen. In this state, when it is present in pepsinogen form, in this state it has an additional polypeptide fragments and its active site. Means ये जो पेप्सिनोजेन है बेसिकली इट कॉन्टेन सम एडिशनल पॉलीपेप्टाइड फ्रैगमेंट्स, और ये पॉलीपेप्टाइड फ्रैगमेंट्स कहाँ पे प्रेजेंट होते हैं? दीज आर बेसिकली प्रेजेंट एट एक्टिव साइट एंड इट डज नॉट अलाउ द सबस्ट्रेट टू बाइंड, सो इट रिमेन्स inactive. This pepsinogen remains inactive. But when this pepsinogen is exposed to HCl, hydrochloric acid, due to stomach activity, now that is the time when the additional polypeptide fragments are removed. ठीक है, यहाँ पे जो पेप्सिनोजेन पे यहाँ पे कुछ एडिशनल पोलीपेप्टाइड फ्रैगमेंट्स मौजूद थे. अब जब क्या होगा कि HCl के साथ एक्सपोजर होगा तो वो पेप्टाइड्स क्या हो जाएंगे? वो रिमूव हो जाएंगे. एंड inactive pepsinogen, which is apoenzyme, is converted into holoenzymes, means it is converted into pepsin. Now we can read this slide. When pepsinogen is first secreted, it has no digestive activity. What is the reason? Because at that time it is inactive. However, as soon as it comes in contact with hydrochloric acid, it is activated to form active pepsin. Now we will discuss the types of cofactor. There are certain types. It may be organic and or it may be inorganic molecules. If we talk about inorganic cofactors, inorganic cofactors get only attached to enzyme when substrate binds. Such cofactors are called activators. Here you can see in this example that glucose plus ATP. Here the enzyme hexokinase is present and it converts the glucose into glucose 6-phosphate with release of ADP and hydrogen ion. So the cofactors can be divided into organic and inorganic. The example of inorganic cofactor, it may include magnesium, iron, copper, zinc. Now we will talk about organic one. Organic molecules are either coenzyme or prostatic group. हमारे पास जो organic molecules हैं उनको हम लोग आगे मज़ीद डिवाइड कर सकते हैं coenzymes में या फिर prostatic group में. Now what are coenzymes? Coenzymes are basically derivatives of vitamins. They have detachable cofactor. Means these cofactors are basically detachable. These are not permanently attached to them. Now here is the example of ethanol. When ethanol undergoes the reaction, here is the enzyme alcohol dehydrogenase. In the presence of NAD, NAD is converted into NADH and this ethanol is converted into acetaldehyde. Example is ATP, NAD and FAD. Again here is a flow sheet diagram in which cofactors are shown. Cofactor is divided into organic. This is organic and this is inorganic. And then these are further subdivided. Now the cofactor. Cofactor, the prostatic group. Now what is prostatic group? Prostatic group is covalently bonded part of an enzyme which permanently attached to the enzyme and does not detach after the completion of reaction. जैसे हमारे पास reaction complete होगा तो वो वहाँ से detach नहीं होगा, but it is permanently attached there. In this image you can see that here is an enzyme and this is basically the active site. Now The prostatic group is permanently attached at this site. And when we talk about coenzyme, in coenzyme, this is enzyme and this coenzyme is basically deattaches when the reaction is completed. Here are some examples of prostatic group. Now here is the here is again a flow sheet diagram. Basically it is a summary. यहाँ तक हम लोग ने जितना किया ये उसकी एक समरी है जिसको एक फ्लो शीट चार्ट के फॉर्म में यहाँ पे शो किया गया है. What are enzymes? Enzymes may be simple one, means they are only protein in nature, or they are maybe complex one. Complex किस तरह से बनता है कि means they have the combination of protein part and non-protein part. What is the non-protein part? That is the cofactor. अब holo enzyme हमारे पास क्या है? Holo enzyme is basically the combination of apoenzyme, means protein part, and non-protein part, which is cofactor. As we have studied that cofactor is divided into two main groups. The one is prostatic group and the other one is coenzyme. Prostatic group are those where it is permanently attached, and the coenzymes, it is loosely bound. Now here is the mechanism of enzyme action, how this process takes place. Basically, in an enzyme's catalase reaction, the substrate first bind to the active site. This is the substrate and it will bind to the active site of enzyme to form enzyme substrate complex. This one. Substrate entering the active site of enzyme. Here the substrate get entry. After the entry, it will convert it into enzyme substrate complex. Then the substrate is converted into product. This substrate is converted into product. As you can see that there is change in color. First it is green in colors, mean it is showing the substrate. But when the enzyme substrate complex, this substrate is converted into product. While it is attached to the enzyme, it will form enzyme product complex. यहाँ पे अब ये क्या बन जाएगा? Enzyme product complex बन जाएगा. And finally the product is released, thus allowing the enzymes to start all over again. Then at this time, here the product will release, and as the product will release, the enzyme will free. ये एंजाइम फ्री हो जाएगा. अगेन इसके साथ एक न्यू सबस्ट्रेट यहाँ पे आके अटैच होगा और उसी तरह से हमारे पास पहले एंजाइम सबस्ट्रेट कॉम्प्लेक्स, देन एंजाइम्स प्रोडक्ट कॉम्प्लेक्स, एंड देन अगेन प्रोडक्ट विल रिलीज एंड लिविंग द एक्टिव साइट ऑफ एंजाइम. नाउ द मैकेनिज्म ऑफ एंजाइम एक्शन. बेसिकली आपको इक्वेशन की फॉर्म में इसको यहाँ पे इस स्लाइड में शो किया जा रहा है कि here is enzyme and here is substrate. When they combine, they form enzyme substrate complex. Then this enzyme substrate complex, substrate is converted into product. Then it will form enzyme product complex, and at the end enzyme and product are released. The product will release and enzyme is free. Here is an animation in which enzyme substrate complexes show. So this is the enzyme, this is substrate, and this is the active site. It combines here, it forms enzyme substrate complex, then enzyme product complex, and then the product is released and enzyme is free for further activity. Again, here is mechanism of enzyme action, but here is an example. This is the example of basically sucrose. Now the substrate sucrose consists of glucose and fructose bound in together. In the first step we have sucrose which basically consists of glucose and fructose, and these glucose and fructose, these are basically bounded together. इन दोनों के दरमियान bond मौजूद है. You can see here that this is the enzyme and this is the active site. In the second step, the substrate binds to the enzyme, forming the enzyme substrate complex. Here it combine and it form enzyme substrate complex. Now water get entry here. Now the third step is binding of the substrate, and enzyme places stress on glucose fructose bond and bond breaks. Here is fructose, here is glucose. इन दोनों के दरमियान क्या है, एक स्ट्रेस वाली कंडीशन आती है, एंड एट दैट टाइम जो इन दोनों के दरमियान बॉन्ड था, दिस वन बॉन्ड यू कैन सी, अब यहाँ पे देखिए वो चीज़ नज़र नहीं आ रही, मीन्स क्या हुआ कि यहाँ पे बॉन्ड की ब्रेकेज होगी, और जब बॉन्ड की ब्रेकेज हुई तो क्या हुआ, ग्लूकोज एंड फ्रुक्टोज आर रिलीज्ड, एंड द लास्ट स्टेप इज प्रोडक्ट्स आर रिलीज्ड एंड एंजाइम इज फ्री टू बाइंड अदर substrate. Now the next is models for enzyme action. There are different models showing how the enzymes are performing, how they are doing their actions. So the first one is lock and key model, and the other one is induced fit model. Lock and key model. Lock and key model was proposed by Emil Fischer in 1894. Now the name of the scientist is very important for your objective question, and similarly the year. So please underline these things. According to this model, lock and key model, active site of an enzyme has definite shape and rigid structure. So particularly substrate will bind to active site. Shape of active site is complementary to the shape of substrate. Lock and key model. What is the reason that it is known as lock and key model? Basically, it assumes that like a particular key opens a particular lock. Enzymes which work according to this model are called non-regulatory enzymes. When one enzyme catalyzes only one substrate and no other, is called absolute specificity. This model is exercised by very small number of enzymes. Here is the example of this model. For example, hydrolysis of urea. Now this urea show highly absolute specificity. How? Like urea and water combines. In the presence of urease, it is converted into ammonia and carbon dioxide. Now this reaction is particular, will take place with this enzyme. Now here is the image which is showing the lock and key mechanism. Shape of enzyme is fixed. In this model, the shape of the enzyme is fixed. In this model, lock and key model basically assume that a particular key, here is a key, opens a particular lock, means the specific enzymes act on substrate. This key, this is the key, means this is the substrate, and this fits at here this enzyme. Like now this is the enzyme, this is the active site, and this substrate come and it will get entry here at the active site. So it will form lock key complex, and if we talk about enzyme, here it will form enzyme substrate complex. Now we further discuss the lock and key model. How this reaction take place. In this reaction, there are 3 steps. In the first step, a substrate is drawn into the active site on the enzyme. Here is substrate, here is enzyme, and it will come to the active site. The substrate shape must be compatible with the enzyme. Now this substrate, its shape is compatible to the enzyme's active site in order to fit and be reacted upon. The enzymes modify the substrate. In this instance, the substrate is broken down, releasing 2 products. Now two products are released. So this is known as lock and key model. Lock and key model is just like puzzle pieces or jigsaw pieces which are fitting together. Now the next is induced fit model, which is proposed by Koshland in 1959. The active site is modified as the substrate interact with enzyme. Enzyme's active site is flexible, so it modifies the substrate. When it modifies as the substrate interact with enzyme, the amino acid which make up the active site are molded into precise shape, which enables the enzymes to perform its catalytic function more effectively. The change which is induced in the shape of the active site is responsible for conversion of substrate into the product. As the reaction is completed, the active site regain its original shape. जैसे ही ये रिएक्शन कंप्लीट होगा तो एक्टिव साइट क्या करेगा, वो अपनी ओरिजिनल शेप पे वापस आ जाएगा. Enzymes which follow induced fit mechanism are called as regulatory or allosteric enzyme. For example, hexokinase. And here is also the example of carbonic anhydrase. Carbonic anhydrase is enzymes which is found in red blood, gastric, pancreatic cell that catalyzes the interconversion of carbon dioxide and carbonic acid. It also play important role in carbon dioxide transformation in blood. Here this carbon dioxide plus water in the presence of carbonic anhydrase, it is converted into carbonic acid, and then this carbonic acid is converted into bicarbonate with the release of hydrogen ion. Here is allosteric enzymes. Now what are allosteric enzymes? यहाँ पे हम थोड़ा सा allosteric enzyme की बात करें कि allosteric, यहाँ फर्स्ट में इस इमेज से मैं आपको थोड़ा सा समझा दूँ कि allosteric means other site. As you can see that there is enzyme and now this is the active site. जैसे कि आपने active site के बारे में पढ़ लिया. अब इस active site से हटके एक और site भी यहाँ पे मौजूद है, and that is known as allosteric site. These enzymes have two receptor sites. The first is substrate fits and the other one is inhibitor or activator molecules fit here. Now we will read from the slide. An allosteric enzyme is an enzyme that contains a region to which small regulatory molecules. Now these small regulatory molecules means effectors may bind in addition to and separate from the substrate binding site and thereby affect the catalytic activity. Here is
Hey guys, quick biochemistry basics here.
A video
about difference between simple enzymes and allosteric enzymes. Simple enzymes only have one active site, whereas allosteric enzymes have an active site and a regulatory site.
Here is
Simple
regulatory
enzymes
site.
are substrate specific, whereas the allosteric enzymes are specific for the substrate and a modulator. In case of simple enzymes, the rate of reaction decreases in presence of competitive inhibitor, while in case of allosteric enzymes, the modulator may increase or decrease the rate of reaction. Simple enzymes shows hyperbolic curve, whereas the allosteric enzymes shows typical sigmoidal curve. Simple enzymes are also termed as non-regulatory enzymes, while allosteric enzymes are termed as regulatory enzymes.
So the induced fit model. In the induced fit model, substrate enters the active site of the enzyme. This is enzyme, here the active site, and here is substrate. It get entering the active site of the enzyme. When the substrate get attached with enzyme, it will form enzyme substrate complex. But enzymes changes the shape slightly as the substrate binds. Enzymes product complex is formed, and after that enzyme is released and product is also released. Basically हमारे पास जो लॉक एंड की मॉडल है उसमें क्या है कि हमारे पास जो एंजाइम की शेप है, जो एक्टिव साइट की शेप है, वो क्या है, दैट इज़ फिक्स्ड, दैट इज़ रिजिड. बट इन इंड्यूस्ड फिट मॉडल उसमें क्या है, उसमें थोड़ी सी फ्लेक्सिबिलिटी है. जैसे वो सबस्ट्रेट वहाँ पे जाके अटैच होता है तो वो उस एक्टिव साइट में थोड़ी सी मॉडिफिकेशन करता है, उसकी शेप में थोड़ी सी मॉडिफिकेशन करता है. उसकी बेसिस पे क्या होता है, हमारे पास एंजाइम सबस्ट्रेट कॉम्प्लेक्स बनता है. और जैसे ही प्रोडक्ट हमारे पास रिलीज़ होता है तो क्या होता है कि ये जो हमारे पास एंजाइम की जो एक्टिव साइट होती है वो अपनी ओरिजिनल पोजीशन पे वापस आ जाती है. हियर इज़ अ वीडियो about induced fit model.
Enzymes are protein molecules that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, is the area where enzyme activity takes place. An enzyme works by binding to molecules, called substrates, at its active site. The job of the enzyme is to convert the substrate into a different product or products through a series of chemical reactions. Following the reactions, the products are released and the enzyme is free to act on another substrate. At the active site, the substrate should fit into the enzyme almost like a key fits into a lock. Because of this, most enzymes can fit only one substrate. To accomplish this lock and key fit, the active site undergoes a slight change in shape in order to better accommodate the substrate. This is called the induced fit model because the enzyme is induced to undergo small changes so that the substrate can achieve optimum fit. All enzymes have optimal environmental conditions that are correlated with maximum enzyme activity. Environmental conditions such as pH and temperature play a role in how efficient the enzyme is in conducting its reactions. When conditions are less than optimal, an enzyme will lose its configuration and slow its activity. This is due to a change in the three-dimensional shape of the enzyme and is called denaturation.
Now, the energy of activation. What is energy of activation? We have discussed it already. Here is the definition: the amount of the energy required to activate the atoms or molecules to a condition in which they can undergo a chemical transformation. हमने जब पहले इसको एक्सप्लेन किया था तो हमने इसको क्या कहा था, बेसिकली हमारे पास जो एक्टिवेशन एनर्जी है, आप इसको सिंपल कैसे, दैट इट इज़ द स्पीड बम्प. ठीक है ना, ये उसको क्या करती है, उसको हेल्प आउट करती है, स्टार्ट करने में उसको हेल्प आउट करती है. द एनर्जी which is added to cause the molecule to react with one another is known as energy activation. In non-living system, we use heat as the energy of activation to increase the number of effective collision between molecules. But in living system, अब living system में इस तरह से नहीं हो सकता. वहाँ पे क्या होता है कि all living cells and organisms are mainly composed of thermolabile protein molecules. Then the chemical reaction is being carried out in the cell at any time. Energy of activation required for such a large number of reactions cannot be provided by the living system. The living system work in isothermal condition, that is the excited state of the molecules. Reactant is achieved by biochemical process. Now, here is the comparison. Comparison when the enzyme is used in a reaction and when a reaction is occurring without an enzyme. Here is reactant हमारे पास क्या है, for example carbon dioxide plus water. If this reaction is taking place in absence of enzyme, at that time it needs a lot of energy to start, to activate, and then after using a lot of energy, then it is converted into product. But if the same reaction is taking place in the presence of enzyme, at that time the activation energy is very low. It will take small amount of energy to start the reaction. Now in this graph you can see that this is here is energy, here is reaction coordinate, here is reactant and product. Now you can say that see that activation energy with enzyme and activation energy without enzyme. Energy of activation. Here is a small explanation. When the activation energy of the reaction is high, it requires a lot of energy to convert substrate into product in the reaction. Hence, only a few molecules of substrate are converted to product. When the activation energy is lowered by the presence of enzymes, the conversion of substrate to product is easier and can happen at faster rate. Here is a video about activation energy.
Enzymes are proteins which catalyze metabolic reactions involving the conversion of a substrate into a product. Enzymes bind with substrates due to their complementary shape. This is called the lock and key model. Rather like a spaceship docking into a space station. In the induced fit model, the enzyme changes shape to accommodate the substrate. Enzymes enable metabolic processes to proceed rapidly at room temperature by providing an alternative reaction pathway of lower activation energy. Without enzymes, these reactions would happen too slowly to sustain life, or would require extreme conditions of pH or temperature. Carbonic anhydrase catalyzes the following reaction between carbon dioxide and water. This reaction proceeds 10 million times faster with the enzyme catalyst than without. A water molecule is coordinated to a zinc atom in the protein. A proton is removed, leaving a hydroxide ion. Carbon dioxide binds to the hydroxide ion on the zinc and the product is released. When a similar shaped molecule competes for the active site, it reduces substrate activity.
Now this is inhibitor. This is related to our next topic. So here is homework. Now what is your homework? That you have to learn and write the properties of enzymes and then draw the diagram of models of enzymes action. Thank you class. Thank you so much.